Okay, so yesterday we talked about all the parts of the cell, what they do, and then we talked about cell transport. Now, we only talked about what I call the big three. Okay, the big three methods of cellular transport are diffusion, osmosis, and active transport. Because those are the ones that the cell uses most often. Of the big three, which is the biggest? Which one does the cell use more than any other? Diffusion. Yeah, diffusion is the most important one. It is the only way that material moves from one place to another within the cell. Okay? Active transport and osmosis only move material across the membrane. Okay? That's great, it gets in and out, but when it comes to moving stuff around within the cell, that is only diffusion. Right? So what you need to be aware of is that on an exam, if I'm asking you a question about one of the methods of cell transport, you're going to need to be able to identify which one it is from my description. Okay, so let's say I'm talking about putting a cell in salt water and asking you what's going to happen to the cell. Okay, which method of cell transport am I likely talking about?
Osmosis.
Yeah, osmosis, because it has to do with balancing salt. Okay, and so that's kind of the identifier for that one, right? If I say that in this situation a cell needs to get materials into the cell from outside, but they need to be in greater concentration than outside, which one am I talking about? Active transport. Okay, so you've got to be able to, figure out which one it is and then tell me about it. Tell me how it works. Okay, things like that. For active transport, that would be, it uses energy, it changes the shape of the transport proteins, it moves stuff against the gradients. Okay, that kind of stuff all got to be in there. All right. Now, we're going to talk about exocytosis and endocytosis. Okay, movement in or out of the cell involving a change in the shape of the membrane. The stuff we have talked about thus far, okay, osmosis and active transport, those don't involve massive changes in the shape of the membrane. Okay, the stuff we're going to talk about now, endocytosis and exocytosis, does actually involve the membrane actively changing shape. Okay, so they're not part of the big three, but they are a way that stuff moves in and out of the cell. It's not something I usually ask about on a written response item on a test, but it could be something that would be a multiple choice type thing. Okay, it's just as long as we understand that the membrane can change shape and either expel material or engulf material. Okay, so exocytosis. So exocytosis is secretion, okay, of large molecules. Right? Now that's the other big thing. Osmosis, active transport, and diffusion deal with small stuff. Okay? And lots of small stuff has to get moved all the time. That's why those processes are most important. Okay? Exocytosis and endocytosis deal with really big stuff. Okay? Stuff that can't go through a carrier protein. Can't just go through the membrane. Okay? It has to actually be taken in and engulfed and packaged. All right? So exocytosis is the secretion of large molecules by the fusion of vesicles with the cell membrane. So this usually results from the Golgi apparatus making one of those vesicles. Okay, so it's either packaging waste or it's packaging stuff that the cell has produced and wants to export. Okay, so that transport vesicle will move towards the membrane. Now, what is that vesicle made out of?
Like in it or outside?
Outside of it.
Lipids?
Yeah. That vesicle is made of the same stuff the cell membrane is made out of. Okay? And that's, so as it's going towards the cell membrane, once it touches the cell membrane, it just merges with it. Okay? It becomes part of the cell membrane, and whatever was inside of that vesicle just pops right out of the cell. Right? So the Golgi apparatus is always making little membranous bubbles, okay, that will go towards the membrane, and when they hit the membrane, they just become part of it, and whatever was in them is now released into the intercellular fluid. Okay, that's the stuff that's kind of outside of the cell. Okay, everybody with me on that? Okay, so that's exocytosis. Exocytosis is a membranous vesicle merging with the cell membrane and expelling its contents outside of the cell, be those waste products or things that were actually built to be exported by the cell. So if we're looking at like any of your glands, okay, like your adrenal gland or your pancreas or your thyroid gland, okay, something that produces hormones, this process is going on all the time because those cells are producing hormones for the rest of the body. Okay, so that Golgi apparatus is constantly producing vesicles that are merging with the outside, okay, and it's constantly secreting, okay, the product it's designed to make. Okay, does that sort of make sense? So that's exocytosis. Okay, endocytosis is the opposite. So if we see this process where the membrane, the membranous envelope kind of merges with the membrane, endocytosis is that process in reverse. Okay, so something touches the outside of the cell and the membrane engulfs it and takes it in. Right, that's endocytosis. There's 3 different kinds of endocytosis. Okay, so in endocytosis, the cell takes in macromolecules. Macro means big. Okay, micro means small, macro means big. Okay, and particulate matter, so chunks of stuff, food, okay, would be in particular, by forming vesicles budded from the membrane. Okay, and there are 3 types of endocytosis: phagocytosis, okay, which is the engulfing of a solid object. Okay, Pinocytosis, which is the engulfing of the intercellular fluid, just cell drinking. Okay, and receptor mediated endocytosis. This is how a cell takes in hormones, okay, and other chemical messengers from the body. Because they're too big, those molecules are too big to just go through the membrane and have to be engulfed. Okay, so the process is the same regardless of which type of endocytosis, phagocytosis, pinocytosis, okay, doesn't matter which, the process is the same, okay, it is simply that the cell membrane kind of pushes in and then kind of pinches off, okay, to make this, this vesicle. So it just buds in, okay, and then re-merges there, okay, so that you now have whatever was in that area engulfed in a sphere, okay, made of membrane material that's now going to move to whatever part of the cell it needs to go to. Right? Okay. So this is how the amoeba eats. Okay? We looked at the amoeba under the microscope the other day. It eats by phagocytosis. That is, that it gets near a food particle and it just engulfs it. Okay? It touches the membrane and the membrane just goes right around it, takes it in. Okay? Then it would merge with a peroxisome and be digested in the same way it would be in our body. Right? It's also how your immune system kills bacteria or infected cells in your body. So your big, like killer T cells, okay, the white blood cells, they'll go around and when they see stuff that shouldn't be there, okay, they'll just engulf it, okay, and then destroy it by essentially eating it. Okay? But they have kind of the special ability to kill bacteria and eat bacteria and stuff that other cells do not. Okay? Is that sort of making sense? Okay. So the first one, like we said, is phagocytosis. Okay, a cell engulfs a particle, so something solid, by wrapping what we call a pseudopod. Okay, that's a false foot. Pseudo means false, pod means foot. Okay, wrapping it around whatever the object is, and then taking it in.
Okay, sorry, so I'm just double checking. So pretty much like the cell has this thing kind of off the side of it? Is that what's happening?
No, any part of the membrane can do this.
Any
Yeah.
part, okay.
Yeah, any part of the membrane. Whatever part it touches, that part would do the engulfing.
Oh, okay.
Yeah. All right. So it does it around it, packaging it within a membrane enclosed sac large enough to be called a vacuole. So the process is very much like if you have a dog, you know, you've ever like had to pick up after them, that's what the process is like. So here's the cell, okay, and if something touches the membrane, okay, the membrane just closes in around it. Now it's inside, okay? It basically just turns the membrane inside out until it's completely engulfed the particle, okay? And now that particle is inside the cell, or your dog's business is inside the bag, okay? Without having to touch it. All right, so that's how phagocytosis works. Okay, questions on it? Yes. All right, pinocytosis, exactly the same process mechanically. The cell just engulfs stuff, but in this case, it's actually engulfing water. Right? If a cell becomes extremely stressed, okay, due to large salt changes in salt balance, it will actively take in water by this same process, but it's called pinocytosis if it's just engulfing fluid. Right? So a bunch of the intercellular fluid will just be engulfed by the membrane, and the fluid will come in these vesicles and then be readily absorbed. Right? This is actually what it looks like under a light microscope. Okay? So you can actually see here, okay, what they've done is just like stressed this cell big time and then put it in distilled water where it just starts going. Okay? And so it's just engulfing all of the water, okay, making those water vacuoles, essentially small vacuoles full of water. Not like the plant cells one, but okay, smaller. Okay, that would just be readily absorbed into the cytoplasm. Okay. And then the last kind is kind of the special kind. It's really the most important of the three, okay? And it's receptor mediated endocytosis, okay, or RME. This is how hormones, which are proteins, which are large molecules, get taken into the cell. So we said the other day that the cell membrane is not this flat, featureless material, okay? It's got all this stuff embedded in it like a Jell-O salad. Well, some of that stuff is receptor molecules embedded in the membrane. And those receptors are like an antenna. Okay? They receive chemical signals, and when that antenna receives the signal, it automatically tells the cell membrane to engulf that stuff. Okay? Engulf it right away and take it in, because that molecule is going to cause something to happen in the cell that's important. All right? So it will take it in very, very quickly through this engulfing process. Right? Now, the receptors don't receive multiple signals. That's why your cell membrane has so many receptor molecules on it. There's a receptor molecule for each signal. It's not like the antenna in your car that can pick up like every radio station. It would be an antenna for every radio station. Right? That's what the cell membrane is like. There's a different receptor. They all have a different shape. Okay? So you can see in this diagram here, the receptor they're talking about is receiving triangular signals. There would be another one that would receive circular signals. Okay? And so there's a receptor for each signal that it would get. Okay? When that receptor is triggered, then boom, the membrane engulfs that material. This is what it looks like under an electron microscope. Okay? So you can see here's the membrane. Okay? And actually on the surface of the membrane, it's easier to see in this picture here, okay? You can actually see these receptors, okay? And the chemical signals that are in them. Right? There's a whole bunch of them here. Okay? Each one there's a whole bunch. I can't draw them all, but okay, you can kind of see them there. When those receptors are triggered, okay, then that stuff, this is exactly what happens. Here's the cell membrane. Okay, it's exactly like the diagram. This is where that's starting to, the process is beginning, okay, and here's where the process is just about done, okay, that's gonna pinch off, and then that vesicle will be inside. Okay, so gone over these. This is endocytosis. That's different than cell transport. I don't want you to get them confused. Okay? Endocytosis is the changing of the membrane to engulf stuff. Okay? Cell transport is diffusion, osmosis, and active transport. Right? All right. I want you guys to answer these 4 questions. I'll give you a few minutes here, okay, to get some answers down for them. You're not necessarily going to find the answers word for word in your notes, and 1 or 2 of them are going to require you to do a little bit of thinking. Okay, but I'm going to give you a few minutes to work on that, and then we'll cover them together. So, color blindness is caused by a person's cells being unable to produce certain proteins. Where do you suppose the problem lies? Keeping in mind that it says certain proteins, not all proteins. Because usually people go ribosomes right away when they say it can't produce this certain protein. If there's something wrong with the ribosomes, it wouldn't be able to produce any proteins. Okay? This is just certain proteins. Where's the problem? Anyone know someone who's colorblind? How does it happen? Don't know. Color blindness is genetic. Okay? It's in your DNA. Okay? The problem lies in the person's DNA. There is a mutation or an error in their DNA, and so the protein that normally allows the cone cells in your eye to perceive certain colors are not producing the right protein. They're producing a protein, but it doesn't work. Okay? The same problem occurs for someone who's diabetic. Okay? A person who's diabetic doesn't produce insulin. They produce a protein that's very close to insulin, but it doesn't work quite right because they have a mutation where on their DNA, the part of the DNA that codes for insulin has a mistake. Okay, so it's the same for color blindness. All right, so the problem lies in the person's DNA.
And how would it work if you got like colorblind, not colorblind, sorry, if you got diabetes from eating too much sugar?
That's a different type of diabetes.
Okay.
Yeah, I'm talking about like the genetic
Okay.
type 1 diabetes.
Yeah, but would it alter your DNA somehow like
No,
that?
it doesn't. Nothing can alter your overall DNA. The DNA of an individual cell could be altered by something that is mutagenic, but you're never going to have a major genetic alteration of your body from anything.
Alright.
Okay. All right. So the color blindness thing, yeah, definitely a mutation, and there's different types of color blindness. Okay. Some people are, almost just barely colorblind, and they don't even realize it until they, have to do their driver's test or something like that, and suddenly they can't tell people what number is in the circle of little dots. Okay. Most people go through their most of their life without even really noticing it. Mr. Van Erve, who subbed for me, he's totally colorblind. Okay. For his retirement gift, we bought him those glasses that allow people who are colorblind to see colors. You should have seen his face. Okay. Guy had never seen green until he put the glasses on. He's like, whoa. Like it was like somebody smacked him in the face when he put them on. Okay? It was just so strange for him to see these things that he hadn't seen before. Okay? Just totally different. And it's just a matter of his genetic makeup is such that his body doesn't produce those proteins that allow the cone cells in his eyes to see color. Okay? It used to be really funny because he would come to school and you could tell if he got out of the house before his wife saw him. Because he wouldn't match at all. One day he came up and we were wearing like the same patterned shirt, but mine was orange and blue and his was purple. He's like, hey, look, we're twins. I'm like, yeah. And we walked down the hallway, look, we're twins. And everyone's laughing because he has no idea we're not twins at all. Okay, and that the colors are, I know, but he was always good with it because he knew. I mean, he knew he was colorblind. It was fun. It's like, hey Jerry, is that a red light or is that a green light? And he's like, it's green. Well, how do you know? You're colorblind. It's on the right. You ever thought there's a reason why stoplights work the way they do? Okay, green's always on the bottom or on the right. Red is always on the top or on the left for colorblind people. Okay, if they don't do that, like if they just got the one light and it's flashing, they're pretty screwed. Okay, how do I know? He would just drive right through that. He wouldn't know whether it was red or green. So yeah, it can be a problem. That's why it's one of the things they test you for when you're getting your driver's license. Okay. All right. Number 2, why is it important for organelles to have their own membranes? Looking for like one word, kind of long. Starts with C. It was super important to highlight it on the very first slide. Kelly.
Oh, no.
Oh, okay.
Compartmentalization.
Compartmentalization. Okay? Each organelle has its own separate set of conditions that help it do its job best. Okay? Compartmentalization. All right, what might happen if the lysosome's membrane ruptured? What'll happen to the cell?
It can lose its enzymes, maybe?
Well, yeah, the enzymes will get out. What do the enzymes do?
Eat perfectly healthy parts of the cell.
Absolutely. Okay? The cell will eat itself to death. Okay? That is in fact how cells, especially in animal cells obviously only, okay, regulate themselves. Cells have an ability to kind of detect when there's something wrong, and if there's something seriously wrong with a cell, it'll take one for the team. Okay? And it'll perform what's called cell lysis. That is, it will rupture all of the lysosomes and the cell will digest itself. Okay? And that way, it's kind of a mechanism by which your body can prevent cancer. Okay? The problem with cancer is it's usually a cell that's so far gone it can't even realize there's something wrong with it. Okay? That mechanism is broken and the cell begins to divide out of control. Okay, number 4, what problems might a cell have if all the ER was removed? This is my kind of backhanded way of asking you what does the ER do? Okay, what does the ER do? Claire?
It digests certain stuff.
Not really digests, it'll break down, there's one thing it'll break down that's food-like.
Well, like for like the smooth ER and stuff, like there wouldn't be like a production of lipids, and then like toxins and like poisonous substances wouldn't be digested or like broken down.
Right.
And then like proteins also wouldn't have been able to like travel or transport as well.
Okay, yeah. So the smooth ER will break down complex carbohydrates. I think that, Claire, that's what you were talking about, break down complex carbohydrates. Okay, the rough ER will transport protein. The smooth ER detoxifies poisons, makes lipids to repair membranes, and fat-based hormones are produced in the smooth ER. All right, so those are all the jobs that would not happen if the smooth ER was removed. So what problems might a cell have? A lot. Okay, yeah. All right, any other questions on those? Okay, so on Monday, we're going to be doing our next lab, okay? It'll be our cell anatomy lab. And in that lab, we're going to be looking at plant cells, animal cells, okay? Those are going to be the 2 most important ones. We're going to make a wet mount of our own cells. Now, because of COVID, we have to be very careful about how we do that part of the lab, okay? You will make a slide of some of your own cheek cells by swabbing the inside of your cheek, okay? Gently smearing it onto the slide and making a wet mount like you did with the letter E. You and only you will handle that slide, okay? And when you are done with that slide, you will throw out the cover slip just like you did with the other one, and then you and only you will wash the slide with soap and water before giving it back to me. Okay? We did this pre-COVID too, but it's the you and only you handling part that's kind of the new part. Okay? Because I didn't want somebody's germy slide back before either. Okay? So that's what you'll be doing. You'll be looking at those 3 slides and making lab diagrams of them, just like we did with this last lab. But it's going to be a little bit tougher, okay? Because we're going to be looking at cells that are pretty small and tightly packed. The stuff we were looking at for the most part was pretty spread out. It was like, oh, well, there's something, that must be an amoeba, or there's something, that's a paramecium. Now, we're going to be looking at a glop of tissue where there's a whole bunch of cells, and we got to try and find a good one where we can locate as many of the organelles as possible. Okay? The purpose of the lab is to figure out what organelles are necessary for basic functions to be carried out. Okay? In other words, what structures are common to all cells as best we can. We kind of know that because we have a cell diagram, but we want to kind of look at it for ourselves. So that's what we'll be doing in this lab. So to give you an idea of what we'll be looking at, okay, we'll be looking at a leaf cross section. Okay, so what we mean by a cross section is if this is the leaf, that's not too bad. Okay, we would be cutting it across like this and looking at this part from the side. Okay, that's what a cross section is. All right, so what we're looking at are essentially the different layers of the leaf, and you can see up here that we have lots of those rectangular looking cells. right? And you're going to be able to see this quite clearly through your microscope. Okay, what do you suppose these little pink things are?
Nucleus.
Yeah, that's a nucleus for one cell, that's a nucleus for another cell. What are these kind of greenish things?
Chloroplasts.
Those are chloroplasts, okay? They're big enough to see, right? And if we look carefully at some of these nuclei, okay, you can actually see that kind of the edge here is a bit fuzzy. The edge of this one here is a bit fuzzy. What would that be? Yeah, rough ER. I mean, I'm not saying use your imagination, but use your imagination a little bit, okay, when you're looking at these. I mean, I'm not saying guess, but make a reasonable assumption as to what things are, right? This big blank area right here, what could, yeah, that's the water vacuole, okay? It's just pushed everything to the side, so it looks transparent, looks like you're looking right through it, okay? That's the water vacuole, right? Can we see where one cell ends and the next one begins?
Yes.
Yep, that'd be a cell wall then, right? Those are the kinds of things I'll be able to see and at least somewhat accurately label, right? Within the nucleus, I can also see the nucleolus, right? So these are all things that I should be able to see and label pretty easily, all right? Okay, when we're looking at the animal cell, okay, we're going to be looking at what's called stratified squamous epithelium. If you've ever dusted your knuckle and peeled it back, it's that red skin underneath that stings like crazy. That's stratified squamous epithelium. Okay, that's what you're going to be looking at under the microscope. All right, now there's a whole bunch of cells here.
Yeah.
Can you see them all? And you're going. Okay, yeah, you can, if you know what to look for. Okay, so right here is an animal cell. See the nucleus?
Yeah.
Okay, and if you look closely, you can see that there's almost like a goatee off this end of it. That would be, yeah, rough ER. Okay, again, not saying use your imagination, but make reasonable conclusions, okay, about what you're seeing, right? And within some of these cells, yeah, there's I can see kind of, other solid particles. This right here is probably the Golgi apparatus for that particular cell. Okay, it's a reasonable conclusion to draw. I can see where one cell ends and the next one begins, so that would be the cell membrane. Okay, I can see the nucleus, so I can see the nuclear membrane. I can see the nucleolus. I should be able to, label a few things at least inside of each cell. Am I going to see everything? Absolutely not. Okay, but I should be able to see kind of the big stuff if I'm doing a good job. Okay, so those two slides are both stained. Right? That means that they've had a dye added to them before they were preserved, which is why we see all the clear colors. Okay? This is another example of that stratified squamous epithelium. Okay? You can really see the cells here, okay, where one ends and the next begins. Okay? So it depends on which one you get, how well lit it is, right? But you'd be able to see it pretty well. This is my cheek. See my cheek cell? It's very nice.
Wow.
Okay. All right. So you got nucleus in here. This over here is probably a Golgi apparatus. This is probably the rough ER right here. Got a nucleolus in the top left of the nucleus. But see how this one's not colored? Okay. Makes it a little tougher to see everything. Right. That's when that little tab on the bottom of the stage called the condenser diaphragm. If you move that, it closes the iris up a little bit, and stuff that's not stained will kind of just all of a sudden jump out at you, okay? Because the contrast will change and you'll be able to see it more clearly. So this is with that almost all the way open, okay? And this is it with it almost all the way closed. Okay, so you can see cheek cell there, cheek cells, a whole bunch. Now, notice that those cheek cells are, somewhat transparent. If you're looking at something that's large and opaque and fibrous, it's food. Okay, because I inevitably have people, their lab drawing is of something they ate for breakfast. Okay, it's big and thick, and they could see it really easily. If you can see it easily, it's not a cell. Okay, finding these cells is a little more challenging. Right, if something is big and dark, it's a chunk of food. Okay? If you had, let's say a whole grain bagel or something, it's a piece of fiber from the bagel. Okay? right? So just be mindful of that. Those are going to be some of the things that we'll be looking for in the lab on Monday, right? So on Monday morning, okay, we'll go through all of the pre-lab stuff. Have the Chromebooks booked so we'll be able to do it without having to use our phones like we did the last time. Okay, lab diagram process exactly the same as before. Okay, but we'll have the whole period basically to just look at 3 slides. So it'll give you lots of time to get a really good picture. Okay, and if you've got it all, then we can come back here and you'll have some time to work on that lab report in class. Okay, questions there? All right. So with that, that's basically the end. By Tuesday, we'll be done the first half of the unit. Okay, the first half of the unit is just about cells. Okay, well, I should say it's like first third of the unit, but it's between the third and a half. Okay, we'll be moving on now to after that multicellular organisms. Okay, and I'll be telling you when your unit exam is probably sometime next week. It is not sometime next week. I will be telling you
Oh, okay. I was
sometime
like,
next week when it is.
No.
Okay. Right, but we'll be looking at probably somewhere around just after Remembrance Day, okay, probably somewhere in there. Okay. All right.
I've seen awesome kids.